Whole blood is composed of plasma and the formed elements. Plasma contains plasma proteins such as albumins, globulins, and fibrinogens. Albumins are responsible for regulating osmotic pressure, globulins are important for immunity, and the fibrinogens are important for clotting. The formed elements include erythrocytes, leukocytes, and platelets, also called thrombocytes. Thrombocytes is a misnomer because platelets are cell fragments of the megakaryocyte, not cells themselves. Erythrocytes contain hemoglobin, which has 4 chains, 2 alpha and 2 beta. Each chain contains a heme molecule, and this heme molecule is responsible for binding oxygen. A rouleau is a stack of erythrocytes. Leukocytes come in nonspecific or innate immunity. Leukocytes come in 2 varieties, nonspecific defenses or innate immunity, and specific defenses or acquired immunity. The innate immunity cells are the neutrophils, eosinophils, basophils, and monocytes, also known as macrophages. The acquired immune cells are called lymphocytes. These include T cells, B cells, and natural killer cells. To distinguish these, neutrophils have 2 to 5 irregularly shaped lobes of the nucleus. Eosinophils have 2 or 3 rounded lobes of the nucleus. Basophils are granulated. Macrophages have a kidney bean shaped large nucleus, and lymphocytes have a rounded large nucleus. Both macrophage and lymphocytes. The mediastinum is the central tissue mass that divides the thoracic cavity into 2 pleural cavities. And in the circulatory system, we have veins and arteries. Arteries carry blood away from the heart, and veins carry blood to the heart, irrespective of oxygen concentration. Capillaries are the site of gas and nutrient exchange. The pulmonary circuit goes to the lungs, and the systemic circuit goes to the rest of the body. The pericardial cavity is a space surrounding the heart. The base of the heart is the top wide portion of the heart, and the apex of the heart is the bottom rounded portion. Atria are thin-walled chambers of the heart that receive venous blood flow from the pulmonary or systemic circuit. Ventricles are fluid-filled chambers that discharge blood into the pulmonary or systemic circuits. Blood vessel walls have 3 distinct layers: the tunica intima, or the innermost layer, the tunica media, or the middle layer, and the tunica externa, also called the tunica adventitia, or the outermost layer. When comparing veins and arteries, we can see that the walls of the arteries are much thicker and the veins are compressible. In humans, the ABO blood typing system is identified by the antigens on the surface of the blood cells. These glycoproteins come in 2 different types, an A and a B type. From these 2 types, 4 blood types can be made: an A with just the A type, a B with the B type, AB with the A and B type, and O with neither type. The body also creates antibodies, or a protein structure that identifies and counteracts specific antigens that are specific to antigens not found in the body. For instance, an individual with the A antigen will have B antibodies. In addition to the ABO system, red blood cells also have an Rh factor, or a rhesus factor, and this is denoted as positive or negative, which indicates the presence or absence respectively. This shows a cartoon depiction of the surface antigens. We see in the type A blood that the surface antigen, that the red blood cell has the A surface antigen and the B antibody, that the B type blood has the B surface antigen and the A antibody. The AB blood has both A and B surface antigens and no antibodies specific to either of those. The O type blood has neither A nor B surface antigens and contains blood with both A and B antibodies. Agglutination is the process of clumping cells together via antibodies that are specific to the antigens on that cell. For blood typing, if the surface antigen is present, the antibody for that antigen will cause agglutination. So agglutination means that the surface antigen is present. For example, if anti-A antibodies are added to A-type blood, it will agglutinate. Conversely, if the addition of anti-B antibodies causes agglutination, it can be said that those blood cells have the B antigen. This process works the same for the rhesus factor. Here's a depiction of all the major blood types in the ABO and rhesus factor system. AB positive has all the antigens, O negative has none of the antigens. So AB positive is the universal recipient because it will have no antibodies to any of the antigens, and O negative is the universal donor because there are no antigens for which serum antibodies will bind to in anyone's body. Understanding the interplay between the antibodies and the antigens is key to understanding how the blood typing system works.